Hello, everyone. Welcome to today's live broadcast, Emerging Role of Liquid Biopsies in NGS for Biomarker Discovery. I am Judy O'Rourke with LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. QIAGEN NV, a Netherlands holding company, is the leading global provider of sample and assay technologies that are used to transform biological materials into valuable molecular information. Sample technologies are used to isolate and process DNA, RNA, and proteins from biological samples, such as blood or tissue. Assay technologies are then used to make these isolated biomolecules visible and ready for interpretation. QIAGEN markets more than 500 products around the world, selling both consumable kits and automation systems to customers through 4 customer classes: molecular diagnostics in the healthcare, applied testing, forensics, veterinary testing, and food safety, pharma, which is pharmaceutical and biotechnology companies, and academia, which is life sciences research. For more information, please visit www.qiagen.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many as we can, and we'll follow up if we don't have time today. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Raed Samara, PhD. Dr. Samara is a global product manager for NGS technologies at QIAGEN with a focus on pre-analytics and targeted enrichment. Prior to joining QIAGEN, he was a postdoctoral fellow at the National Cancer Institute, conducting research in the field of cancer immunology with emphasis on identifying strategies to boost the efficacy of cancer vaccines. He received his PhD from Georgetown University in tumor biology. I will now turn it over to Dr. Samara for his presentation.
Thank you, Judy, and good morning or good afternoon or good evening, everyone. Welcome to today's webinar on emerging role of liquid biopsies and NGS for biomarker discovery. So just by way of agenda, these are different topics that I would like to cover in today's webinar. First, I'm going to go through a background on liquid biopsy for precision medicine. The next topic would be addressing the challenges that are associated with biopsies and how we could use them for precision. And then the third topic would be how those liquid biopsies are used. I'll cover an example from the literature that have used NGS for studying some mutations through liquid biopsies, and then close this webinar with a summary and questions. So on to quick, brief background on liquid biopsies for precision medicine. So when we think of precision medicine, we think of the right drug for the right patient at the right time and at the right dose. This is in contrast to what traditionally has been done for disease management, which is the one-size-fits-all approach. We know that the one-size-fits-all approach does not work for everyone. So for instance, if you look at the on the left-hand side, I'm showing you sort of a brief description of what a one-size-fits-all approach would look like. So you have a patient is going to be prescribed a certain therapy. However, because we do not really know what the genetic background of that patient is, that patient might, you might see positive effects, you might see no effects at all, or you might see adverse effects. With the precision medicine approach, the idea is to use the genetic information from these patients in order to prescribe certain therapies that will only give you some positive effects. And one of the ways by which we can obtain this genetic information is by liquid biopsy. So a liquid biopsy is basically a biopsy that we would take from fluids. This could be blood, it could be cerebrospinal fluid, it could be urine, it could be any type of liquid liquid form that we can get from an individual. And again, this is in contrast to solid biopsies, which are invasive and would take a lot of time and discomfort to the individual. So liquid biopsies are a great way, are convenient way to obtain this type of information. So what types of genetic information can we get from individuals? What I'm showing you here are some DNA variants that have been used for precision medicine. There are a lot of DNA variants that can exist in any one of us, but the three heavily studied types of DNA variants are point mutations, as you see on the far left. So with point mutations, you get a change in a nucleotide. And this change can actually result in a constitutively active form of a kinase, for instance, that will result in the development and progression of cancer. Another form of a variant is the indels, insertions and deletions. So this is where you have certain nucleotides being inserted into a string of DNA, or some nucleotides being removed in the form of deletion. And then you also have copy number variants or variations. And with copy number variations, you get basically a duplication or triplication or so of a specific gene. So these are the three sort of heavily studied types of mutations, but there are a lot of other mutations like structural mutations or structural variants that could exist, but I'm not going to touch upon those today. So why is this information important? This information, or knowing this information, why is it important? This information is very useful because it can tell us a lot about the disease. And my focus today is going to be a lot on cancer, just because that's a topic that is dear to my heart. So when we look at what, those different types of mutations as they accumulate in cancer patients, while those mutations are detrimental in the sense that they actually result in the development and progression of cancer, we can actually, to our advantage, we can use that mutation and develop and target that specific mutation and obtain those positive effects that I talked about earlier. So, for instance, if we look at melanoma, one of the driver mutations in melanoma is this BRAF V600E mutation. This is a mutation that is found in maybe more than 50 or 60% of melanoma patients. So while this mutation is actually going to drive the development and progression of melanoma, we can actually target this specific mutation with a drug called vemurafenib. So vemurafenib was developed to specifically tackle those melanoma cases that have these V600E mutations. An example of how we can use insertion and deletion information to our advantage for precision medicine is the EGFR deletions in lung adenocarcinomas. So a lot of tyrosine kinase inhibitors have been developed to tackle these types of DNA variants. And then finally, for copy number variations, the best example that I can think of for copy number variations is HER2. So HER2 is amplified in certain forms of breast cancer. And again, while this amplification is going to drive the development and progression of breast cancer, its presence or the duplication of or amplification of HER2 can actually be targeted by a monoclonal antibody that has been developed, and it is called trastuzumab or Herceptin. So again, while these types of variants are detrimental because they can lead to the development and progression of cancer, we can actually now develop drugs and target those specific mutations and achieve those positive effects through precision medicine. So how many actionable DNA variants are there? And what I mean by actionable DNA variants are those DNA variants that we can take action against. Again, the example of the BRAF V600E mutation. If that mutation is present, we know we can target it with that specific drug. But if we look at the total number of actionable DNA variants, how many are there? I would argue that there are only a handful of DNA variants. So you might end up with, for a specific disease, maybe tens or so of these actionable DNA variants. We all have DNA variants in our DNA, whether you're regardless of the ethnic background or so, there are a lot of variants that exist within us. So you have all these common variants, right? And then you have a lot of variants that might develop, but they do not have any obvious effect on a protein. So they're just consider them like passenger mutations. We don't really know what they do. And then if you go through that filtering process and end up with those mutations or variants that are not common, that seem to have an obvious effect on a protein, but then you still have to study those types of variants and understand what they do from a biological and clinical perspective through this iterative biological interpretation step. So once you study several variants, you might end up with only a very handful number of variants that, through biological interpretation, actually show that they can contribute to the development and progression of cancer. So after you go through all these filtering processes, what you end up with is probably only tens of biologically relevant and actionable variants. And again, examples of those variants were the BRAF V600E mutation, the amplification of HER2, and so on and so forth. So there's really a very limited number of DNA variants that we can act upon. So just to give you an example, so this is data that I adapted from a paper that was published in Cell back in 2012, and it looked at the current lung cancer biomarker landscape. So when I say current, that goes back to 2012, but however, this landscape has changed much. So in this paper, what the authors did, they wanted to understand the types of mutations and the percentage of these mutations that accumulate in lung cancer patients. And so what you see here is just the percentage of patients showing these specific types of mutations. So for instance, the authors show here that 25% of lung cancer patients accumulate mutations in the KRAS gene. And then you have 23% of accumulating mutations in the EGFR gene, and so on and so forth. And then you have 37% of them with an unknown mechanism, right? So you have still a large number of patients where we do not really know what the cause is. Now, but the point is, if you look at the types of or the number of genes, you might end up with maybe 5 or 6 or 7 of them for lung cancer when we look at the lung cancer biomarker landscape. So again, a handful of genes that are important for this specific type of cancer. Now, this information is important because it allows us to understand the underlying mechanisms of lung cancer, but it is also important from a therapeutic perspective. So when those are at patients with a mutation in EGFR, known as the L858R mutation, the authors show that these types of patients with this mutation, they achieve a good response rate when they are treated with tyrosine kinase inhibitors. So these are the erlotinib and gefitinib tyrosine kinase inhibitors that I mentioned earlier. So you have a patient or individual with this type of mutation. They get prescribed these tyrosine kinase inhibitors. You see a really good response rate. However, this response rate now drops if the same individual has a mutation in the KRAS gene in the form of G12C. So the addition or the presence of this one mutation only is going to now drop the response rate to these tyrosine kinase inhibitors, showing you that it is still very crucial to understand what types of mutations. If we really want to take advantage of the precision medicine approach, we really need to know what types of mutations are present in these individuals. And that knowing these mutations does not help us understand the underlying cause of this disease, but it's also going to help us in managing this disease. And in the example that I'm going to show you from the literature later on, you'll also see how additional types of mutations can mediate resistance to therapy. So again, it's not only to understand the disease, not only for prescribing therapies, but it also helps us understand also how resistance to certain therapies can develop in these patients. So, but then the question is, how do you test for these mutations? There are two major ways to test for these mutations. You can test for them sequentially, or you can test for them in a parallel manner. Sequentially means that you test for KRAS mutations first, for EGFR second, for BRAF mutations third, and so on and so forth. Now, while a lot of these approaches are very sensitive, the amount of time it takes if you want to do sequential testing is a long time. I mean, if you think of a day for KRAS testing, a day for EGFR testing, it could take you a week or so if you want to go through all of those mutations that are known to contribute to lung cancer. This is as opposed to parallel testing. With parallel testing, you're looking at all of these mutations at the same time. And that is where the power of NGS comes in, because we could use now a technology that allows us to look at all of these mutations, not just in one sample, but also in a lot of samples simultaneously. It cuts down the cost and time associated with these types of testing. So my focus next is going to be on NGS and how NGS, or next generation sequencing, can be used for the testing of all of these different mutations in liquid biopsy samples. Now, the idea of liquid biopsy for precision medicine has existed for a while. This is the one paper that I can think of that really took advantage of a liquid biopsy approach as a non-invasive way to identify and monitor cancer mutations by NGS. It's a paper that was published in Science Translational Medicine back in 2012. And what the authors here from the UK did is that they used liquid biopsy to accomplish three major objectives. First is to screen for and identify low-frequency mutations as low as 2%. So when you think of cancer samples, you always think of heterogeneity associated with these samples. Heterogeneity means that you will find you won't end up with a homogeneous population of cells that share all the genetic information together. What you end up with is a mix of cells that have, and each one of these cells has its own sort of genetic signature, and that's what I mean by heterogeneity. So when you look at the sample in its entirety, the driver mutations, because they're not going to exist in all of the cells because it is a heterogeneous population, those driver mutations might end up being 2% or 3% or even 1% of your sample. So that's what I mean by low frequency mutation. And the second objective was to non-invasively identify the origin of metastatic relapse in a patient with multiple primary tumors. So again, they use that information to understand the underlying mechanism of the development of the disease. And they also, the third objective, they wanted to monitor tumor dynamics by tracking 10 mutations in plasma of a metastatic breast cancer patient over 16 months. So again, this shows you the power of using a liquid biopsy approach. With liquid biopsies, you can take, I don't want to say as many biopsies as you like, but it is much easier to obtain a liquid biopsy than to obtain a solid biopsy. So you can actually use that type of biopsy now to monitor disease progression much more effectively and efficiently than by using the solid biopsy approach. So how can you examine those genetic variants by NGS? There are three very high-level sort of levels of NGS. You have the whole genome sequencing approach, you have the whole exome sequencing approach, and then you have the DNA sequencing approach. Now, which level should we use? To think of the different approaches the way you would see it first. So in the whole genome approach, you're getting a very high level. So think of yourself as if flying a helicopter or a plane, and you're just looking at the very top of this forest. With a whole exome sequencing approach, what you're trying to do is to, instead of getting this very high level view, you're getting a high level view, right? So instead of just seeing the top of those trees, now what you're starting to see more detail about that forest. You have to see the trunks of those trees, not just the leaves. With a targeted DNA sequencing approach, you're actually looking at how those trunks look. Okay, so you're getting a deeper view of that forest, and with targeted DNA sequencing, you're getting a deeper view into that DNA sample. Now, how do they differ? So what I've tried to do here is to compare the three different sequencing levels on different parameters and attributes. So first, on information level, with a whole genome sequencing approach, you're looking at everything, right? So you're looking at every single base that exists in that DNA sample that you're studying. With a whole exome sequencing approach, you're looking only at those regions that code for genes. So you shrunk your genome down to those regions that will code for protein. With a targeted DNA sequencing approach, you as a user or a researcher, you're going to define what regions you want to look at. Those regions can be, they can be genes sequencing certain exons of genes. They can be intronic regions, promoter regions, however you as a researcher, however you define that sort of information level. So it is much more tailored to what you want to do. Now, when we look at cost per sample, the three levels differ dramatically. While we are all striving for that $1,000 genome cost, we're not there yet. So when you look at the cost of doing a whole genome sequencing on a sample, it still costs about $5,000. So for all numbers in the U.S., I know that you know, they will vary depending on where you are, but I would say the numbers are fairly close to each other. So we're still at the, you know, more than $1,000 for a genome. And even when we look at an exome a sequencing experiment, it still costs a lot of money. It would cost about $2,000 if you want to just sequence the exome in that sample. With a targeted DNA sequencing approach, you can drop that cost down to maybe $200 or so, simply because you are narrowing down the regions that have to be sequenced, and by narrowing down those regions, what you could do is you could multiplex many samples simultaneously, which means you can actually sequence many samples for defined all in the same run. So think about it this way: if you are spending $1,000 or $2,000 for sequencing reagents on a sequencing platform, but you are able to analyze 100 samples simultaneously, you take that $22,000 and divide it by 100 samples, so your cost per sample is only $20. So that is really the power of NGS also. It's just the ability to look at many samples simultaneously and examine many different mutations at the same time. So the other attribute is the detection of low-frequency mutations. So remember, I mentioned this earlier when I talked about the low-frequency mutations and heterogeneous samples. So again, when you're dealing with these types of heterogeneous samples, you are going to be looking at some mutations that are present at low frequency. And it is important to detect those types of mutations because a lot of those low-frequency mutations are the ones that are driving the progression and development of cancer. Now, so the question is, which sequencing level can you use in order to detect those low-frequency mutations? With a whole genome approach, the depth of sequence that you get from a whole genome approach is not going to be enough to look at those low-frequency mutations. With an exome sequencing approach, maybe, you might get enough depth to examine some of these types of mutations. But with a targeted DNA sequencing approach, again, because you are restricting the sequencing to certain genes or certain regions in the genome, you're getting a lot more depth of coverage. What I mean by depth of coverage is just the number of times each base is sequenced. And this is extremely important if you're looking at low-frequency mutations. You want to get as high as coverage as possible in order to accurately examine those low-frequency mutations. So with a targeted DNA sequencing approach, yes, you can look at these low-frequency mutations. Now, if I look at also the DNA input, so how much DNA do you need, and also the number of samples multiplexed, you see a huge variation between, or differences between these three approaches. For DNA, if you're using a whole genome sequencing approach, you need 1 microgram or so. So this could be limiting for some samples, like FFPE samples. If you're trying to analyze genetic, the DNA isolated from FFPE samples, you might not be able to do a whole genome sequencing approach. With a whole exome, this drops down to between 100 to 200 nanograms. But with a targeted DNA sequencing approach, you can go as low as 10 nanograms per reaction. So you're using a lot less DNA, which actually saves you saves most of that sample if you want to use it for other studies. And it also facilitates the analysis of those samples that do not yield a whole lot of DNA, like a liquid biopsy. I mean, you're not going to get micrograms of DNA from a liquid biopsy. You might get 10 nanograms, 20 nanograms or so, depending on whether it's a healthy individual or a diseased individual. So the amount of DNA drops dramatically. Also, the number of samples, when we look at the number of samples that you can multiply, again, this is the number of samples that you can run simultaneously in the same reaction. With a whole genome approach, you might, I don't think you can do 2 in the same reaction. So all of that cost, $5,000, is going to be used for only 1 sample. When you look at a whole exome sequencing approach, you can maybe multiplex 2. When you look at a targeted DNA sequencing approach, you can multiplex up to 96 or even more, depending on how many samples you have and what kind of depth you're looking for. But the idea here is that you can run many samples simultaneously if you're using a targeted DNA sequencing approach, which again drives your costs down. I'm seeing some questions come in. Just for the sake of time, if you can hang on till then, I will get to all of these questions later on. Thank you. So what are the benefits of targeted DNA sequencing? It gives you more relevant data because you are defining the regions that you want to look at. It is more cost effective because you have the ability to do this higher multiplexing approach, your costs are going to be decreased. And then it's the ability to detect low-frequency mutations. Again, these are mutations that could be extremely important from a biological and clinical perspective and lower DNA requirements. Okay, so why choose a targeted DNA sequencing approach? It gives you well-defined content. So you as a researcher, you're defining the content. You can shrink that genome to smaller target sizes. This allows you to multiplex many samples to save money. And it gives you more reads per sample, so it gives you more coverage, and this allows you to detect low-frequency mutations. So just to kind of put this into in a graphical format, the idea is to again shrink that genome down to those genes that you are interested in. So if you're interested in studying only KRAS, EGFR, and IDH1, it makes sense to use a targeted enrichment approach to shrink that genomic DNA that you isolated from a liquid biopsy or any other sample. It makes sense to shrink that down to only those 3 genes, and so that when you do your analysis and sequencing, you're only sequencing and analyzing those 3 genes and looking at the mutations that are present in those 3 genes. Again, this is an approach that can be extremely helpful if you have a lot of samples, or if you do not want to know a whole lot about your sample, right? So again, this goes back to the number of DNA variants, actionable DNA variants that are present. There are a lot of variants whose function we have no idea what it does, right? So there could be a mutation in I don't know, X gene or gene X that has been seen in a couple of cases, and we really do not know what it does. So if you're only interested in looking at the actionable DNA variants, the targeted DNA sequencing approach can be a very, very useful technique. So what are the challenges associated with a liquid biopsy? There are several challenges. The 3 main ones is the low DNA quality, the low DNA quantity, and the low variant allele frequency. I kind of alluded to those in previous slides, but I just kind of want to dig deeper into those challenges in this content now. So looking at the low DNA quality, we know that the DNA that you get from liquid biopsies is heavily fragmented. So you're not getting intact DNA. You might have bits and pieces of DNA floating around. And because it is highly fragmented, you want to restrict the way you sequence those types of DNA to short library fragments. Now, there are 2 ways by which you can do the enrichment part. You can do multiplex PCR or hybridization-based approaches. We wanted to take advantage of the high specificity and uniformity of PCR, so we developed a multiplex PCR-based approach. It takes advantage of the specificity and uniformity of PCR. And this was actually there was a nice comparison between the different targeted enrichment approaches. It was a paper published in Nature Methods back in 2010, and it nicely compared the different targeted enrichment approaches that you could use if you want to enrich for specific regions or specific genes in your DNA. And it basically, one of their conclusions was that the specificity and uniformity of PCR cannot be matched by any other method, and that was the basis for our products. We wanted to take advantage of PCR because of the high specificity and uniformity. And the way multiplex PCR works is actually very simple. Let's take an example here for gene 1 and gene 2. Let's call those KRAS and EGFR. The idea is to design primers against KRAS and EGFR. as you see in those arrows. So these arrows just represent primers that have been designed to allow you to amplify EGFR and KRAS. These could be primers against all the exons in these two genes, or it could be they could be primers against certain regions within those two genes. And then you take those primers and you just split them into an appropriate number of PCR pools. That could be 1, 2, 3, or 4. Again, depending on how many primers you have and how far apart those primers are that have been designed to target the regions you're interested in. So again, take those genes, design primers against those genes or those regions, and then split them into an appropriate number of PCR pools. That is the idea of a multiplex PCR-based approach. But of course, you have to develop also the associated PCR chemistry that allows you to amplify those two genes or regions. So that, looking at multiplex PCR, it is just primers against certain genes. Now the trick is by how do you design those primers? Because if you think of this approach, you might end up with thousands of primer pairs in the same reaction, right? So primer design is an extremely important parameter to think of when you're looking at multiplex PCR-based approaches. So, but again, the idea is that you take that genomic DNA, you mix it with the primers and the PCR chemistry, and what you end up with are these double-stranded amplicons that represent your genes of interest. So again, going back to this example of KRAS, EGFR, IDH1, if you're only interested in these genes and you want to use a multiplex PCR-based approach, what you do is design primers against all of these three genes, and then take those primers, mix them with the genomic DNA. This is unfragmented or unprocessed genomic DNA in the sense that you do not need to fragment it anymore. It's already fragmented because it's from liquid biopsies, right? So you take those primers, mix them with the genomic DNA and the PCR chemistry, and what you end up with are these amplicons, like these double-stranded pieces that you see in red, purple, yellow, and green. So these now amplicons represent your genes of interest. So the way we have developed those amplicons is to generate, we wanted them to be around 150 base pairs in size. This is a small size compared to other larger fragments, and the reason why we wanted to keep them small is because of the fragmented nature of the liquid biopsy, of the DNA that you get from liquid biopsy. If you use longer amplicons, these longer amplicons might not work efficiently with fragmented DNA. So again, because of that challenge, you have to have a solution that allows you to generate small amplicons. And that's what we have done with the gene read panels that we have developed using multiplex PCR-based approaches. So low quantity. So you're not going to get micrograms of DNA from your sample. So you need a solution that allows you to use, very small amounts of DNA for target enrichment. With the solution that we have developed for targeted DNA sequencing, you're only using 10 nanograms per PCR pool. So remember that you take those primers and split them into an appropriate number of PCR pools. If you end up with, let's say, 2 pools, you need 10 nanograms each, so you need a total of 20 nanograms of DNA, which is something that if you're looking at liquid biopsies from sample patients, that is something that you can easily achieve. Now, the other challenge is the ability to look at low variant allele frequency. So again, this goes back to the idea of a heterogeneous samples that I mentioned previously. You're looking at mutations that are not every single cell. So what you end up with in your sample is just DNA mutations that are present at low frequency. Now, in order to address this challenge, what we have done is we've taken sort of unique amplicon designs or design approaches. So what I'm showing you here is the way our amplicons would look like if you're specifically looking at, let's say, the G12 locus of KRAS. So this is a locus that is heavily mutated in cancers, and in order to make sure that you see that specific locus in your samples, the way we've designed the amplicons, if you look at those blue bars here, these blue bars represent amplicons that will be generated from the actionable mutations panel, for instance. You see what we call a deep or redundant tiling approach. So you see a lot of amplicons that cover that specific locus. And the idea is we want to ensure that specific locus gets covered in your samples And the way we do this is by designing multiple amplicons. So in case one of the amplicons drops out for whatever reason in the PCR reaction or in the PCR setup, you still have 3 amplicons covering that specific locus. So we need to ensure that those actionable mutations that I talked about earlier are actually covered in your liquid biopsy. So another important feature to, or challenge actually, for liquid biopsies and looking at amplicon sequencing is the ability to protect the ends of amplicons. So when anyone was sequencing will realize that the last 2 or 3 bases of an amplicon do not actually get sequenced very efficiently. It's just the nature of the sequencers that we have currently on the market. In order to protect those ends, what you need to keep the primer sequences at the ends of those amplicons in order to protect those last 2 or 3 bases of your amplicon. So by doing so, by keeping the primer sequences and actually sequencing those primer sequences, what you end up missing are the 2 or 3 bases of the primer sequence itself. And these are sequences that you will not be using anyways for your variant calling step. You will only be using this for mapping and alignment. So by keeping the primer sequences at the ends of the amplicons, you are actually protecting the ends of those fragments that you want to sequence, right? And this is extremely important because you end up with less negatives. Again, if you think of the situation where you do not have primer sequences, you will end up missing those 2 to 3 bases of that fragment. And if there are important variants present in those at the ends of those fragments, you're not going to be able to see them, resulting in false negatives. So this feature is extremely important if you're looking at low frequency mutations, and this was actually very nicely described in a paper published I think back in 2014, yes, in BMC Genomics, and it specifically addresses this this concern. And there was another paper that actually looked at also the benefits of having or keeping the primer sequences at the ends of amplicons. It's a paper that was published this year in BMC Cancer, and they basically, what they did, they wanted to compare 2 different approaches for multiplex PCR. One that digests off the primer sequences from the amplicons, and then the QIAGEN approach, which does not digest off the primer sequences. And when they the results in terms of sensitivity and specificity of those assays, they were seeing that the other multiplex PCR-based approach is delivering 93% sensitivity, whereby 5 mutations out of 74 are missed. And the reason those 5 mutations were missed is because they were present at the amplicon boundaries. So again, these are the 2 or 3 bases at the ends of those amplicons that will be missed if you do not have primer sequences. Okay, so another challenge is the number of, or the different types of sequencers that exist on the market. So you know, if you look at Illumina has a lot of sequencers on the market. Then you have the Ion Torrent sequencers and other sequencers as well. So having all these different options complicates the sample processing steps. So what we have done here is to have this universal approach. So the same panel can actually be used on different platforms, whether it's a platform from Illumina or a platform from Ion Torrent. So it's a platform-independent solution. The types of panels that we have developed vary, but they address different applications, from solid tumors to hematologic malignancies, to tissue-specific approaches, and even gene-specific approaches like BRCA1 and BRCA2, and even a more comprehensive approach like the Comprehensive Cancer Panel. The content of these panels comes from different databases like the Cancer Genome Atlas, from NCCN, from COSMIC, from Nature Reviews, from even private databases like the Ingenuity databases. So we've drawn the content from all these different databases for, you know, and put all that content into specific panels that make sense for different applications. Now, if you do not like the content for whatever reason, you can build your own customized panels. There are 2 ways to build these customized panels, either through the mix and match offering or the custom offering. Through the mix and match offering, you get access to all those gene designs that we have tested in-house. There's about 570 bench-tested gene designs, so you can build your panel from those 570 bench-tested gene designs, or if it's something that does not exist within those 570, you can go through a custom offering, and that way you basically build, the panel that is specific to your needs. And again, through this custom offering, you can build panels for genes, you can build panels for exons, for introns, for promoter regions, any region that you can think of, as long as it is well annotated within the human genome. Okay, just to kind of give you an example of the power of mix and match, these are gene designs that we have tested in-house. Those gene designs are going to behave similarly regardless of the panel that they exist in. So if I look at the gene design for the MET gene, it is present in 4 different panels, from the tissue-specific panels to the more comprehensive cancer panel. And when we look at the specificity and uniformity of those of the MET gene design, we know that it is delivering high specificity and uniformity regardless of the panel in which it exists. Again, because these gene designs have been optimized, tested in-house. So just for the sake of time, I think I've mentioned all of these features previously. Or the one thing that I did not mention is that you end up with one library per sample. So in our workflow, we pool all of our amplicons coming out of a sample after the enrichment step. So going into library construction, you only end up with one library per sample, as opposed to ending up with multiple libraries per sample, which saves a lot of time and saves a lot of money. Okay, for data analysis, just to briefly talk about the data analysis, because this is also a major challenge with NGS and liquid biopsies. There are several tools that exist out there through QIAGEN. You can get access to a lot of databases and a lot of solutions through the CLC Bio offerings for analysis of NGS data, and then the Ingenuity databases, as well as BioBase, if you want to look at if you want to interpret what those variants mean. So you've detected certain variants in your sample. What does it mean from a biological and clinical standpoint to see those variants? So I want to skip those for the sake of just to kind of quickly go through this. The CLC Bio pipelines allows you to build your own algorithms by constructing these different windows. So you define how you want to map your reads, you define how you want to call the variants, and so on and so forth. And for ease of use, there is a plugin for the GeneRead DNA-seq panels that I mentioned, just to kind of make amplicon-based analysis much easier. The Ingenuity databases is a really powerful tool to allow you to, again, make sense of that data. So again, you've seen those variants. What does it mean from a biological and clinical standpoint? Now, so when we think of the targeted DNA sequencing approach, we see it heavily being used in clinical and translational research. And again, for these different types of research areas, you do need different content. So for the clinical research, where you want a high degree of detection, we've developed these very small panels, the actionable mutations and clinically relevant panel, to allow you to achieve this very high degree of detection and allow you to also multiplex many samples together. If you're interested in the translational side of research, the more comprehensive panels make more sense because they allow you to do some discovery. The ability to detect lower frequency mutations drops compared to the smaller panels, but also it allows you to do some discovery. And because the target size is a little larger than the actionable mutations and clinically relevant panel, your ability to multiplex drops as well. Now, the panels, although you are restricting your area of interest, you can still use those types of panels for mutation discovery. Here's a paper that was published in Nature Genetics back in 2014. It actually used a very small panel, but that panel facilitated the identification of a new BRCA2 mutation, the 3'9976 mutation. So you can still use smaller panels to do some form of discovery. And also, the clinical value has been questioned. The clinical value of a targeted DNA sequencing approach has been questioned. However, there are multiple papers out there that actually show you that it's extremely important to look at those low-frequency variants by a targeted DNA sequencing approach. This is a paper that was published in 2014 by in the Journal of Clinical Oncology, and they actually only used a small sample to look at BRCA1 and BRCA2 mutations, and they say that the results that they received from that small panel prompted consideration of a change in care. So again, you can still use a focused type of approach to for disease management. So just to kind of give you an example of how a liquid biopsy approach can be used, I picked a paper that was published in Nature Medicine. But just to give you a little bit of background about the paper and their approach, so they're focused on lung cancer, or the authors are focused on lung cancer, and we know that EGFR mutant lung cancer. So these are lung cancer specimens that have mutations in EGFR. We know that they are sensitive to those EGFR tyrosine kinase inhibitors, as I've shown previously. However, over time, we know that there are a lot of mutations that will accumulate that mediate resistance to these tyrosine kinase inhibitors. And just as an example, this is data from a paper published in 2011, and it actually looked at the mechanisms of resistance to these tyrosine kinase inhibitors. And the commonly seen mutation that mediates resistance to tyrosine kinase inhibitors in lung cancer is this T790M mutation in EGFR. So what researchers have done is they've developed certain drugs that target this T790M mutation. The idea is if you see this mutation being developed as a resistance to the first class of tyrosine kinase inhibitors, why not develop a second class that specifically targets T790M? And this has been a very successful story. There are multiple drugs now that target this type of mutation for lung cancer with some very good results. However, knowing how cancers behave, most likely they will develop even more resistance mutations, resistance to that second class of tyrosine kinase inhibitors. So with this paper, the one that I mentioned earlier, the authors wanted to answer this question. Can we identify potential mechanisms of resistance to AZD9291, which is a drug that is targeting the T790M mutation, before the availability of resistance biopsy specimens? Which means, can we identify potential resistance mechanisms before we can actually get a solid tumor, before we actually see those resistance develop in the form of another solid tumor? So what they wanted to do is to take a liquid biopsy approach, and the paper, again, was published in Nature Medicine in May. So they wanted to take a liquid biopsy approach using the gene-read targeted NGS panels on cell-free DNA, and these were this were samples that were taken from a phase 1 clinical trial. The idea was, okay, well, can we take just blood samples, process them, and sequence the DNA that we get from those blood samples, and look at what types of mutations accumulate in these patients, and try to identify mutations that could mediate resistance to that second class of tyrosine kinase inhibitors. So their approach was fairly simple. They collected blood, they prepared plasma, they isolated cell-free DNA using the QIAAMP circulating nucleic acid kit. They enriched 20 genes. These are genes that are heavily implicated in lung cancer using the gene-read DNA-seq lung cancer panel. They constructed libraries for, okay, do you still see me? I don't know if we lost the connection. I got a note that there was a failure to reconnect, but I'm still getting
no, you're doing fine.
We're good. Okay,
so
okay, yeah, great. So they constructed libraries for a HiSeq, and then they did this ultra-deep sequencing, getting up to 30,000x, which means that all those genes, all those 20 genes were sequenced 30,000 times, basically, right, using this HiSeq. This is a powerful sequencer, so it allows you to actually get up to that level of coverage. And again, this type of coverage is extremely important if you're looking at low-frequency mutations. You might not need to go up to 30,000x, but because they're using liquid biopsies, they wanted to ensure that every single base in there gets sequenced many, many times. Okay, so what did they see? So as a proof of concept, they just wanted to look at plasma samples at baseline and progression. Baseline means, when the patients first entered these clinical trials, and progression once they started seeing some resistance against these tyrosine kinase inhibitors. So when you look at the types of mutations, they looked at the T790M mutation, which is present in baseline and under progression conditions, because those patients were known to have this T790M mutation, right? So the drug that they are trying has been developed against T790M mutation. So you expect to have this mutation in there. However, when the researchers looked at samples under progression conditions, they started seeing this C797S mutation that they saw under progression conditions, but they were not seeing this at baseline, which means that this mutation has actually developed over time after those patients were treated with this second-class tyrosine kinase inhibitor. So this was just as a proof of concept. They wanted to identify what mutations they could be seeing in patients that develop resistance to that second-class tyrosine kinase inhibitor. And to take it a step further, they screened many more samples and started looking at the mutations that are present in the tumor itself and in the plasma samples. And again, they were comparing baseline and progression. So again, in the tumor and plasma, you still see this T790M mutation. Again, something that's expected because these patients already have this mutation. Now, when we started looking at the other mutations that can develop, specifically the C797S mutation, they started seeing this mutation accumulate under progression conditions in both the tumor and the plasma, which, and the idea here was can the plasma samples accurately tell you what is happening in the tumor? So for the C797S mutation, it clearly tells you what is actually happening in the tumor, and it could be used as a surrogate to look at the mechanisms of resistance in the tumor itself, because you see now the C797S mutation in the plasma samples under progression conditions as you see it in the tumor samples under progression conditions. And not only did they see this T2A mutation that results in the C797S mutation, but they were also seeing some other types of mutations in the same locus that will result in this C797S mutation, but it was a different form. So instead of seeing a T2A change, they were seeing a G to C change in the same locus. So not only are you seeing changes in the amino acids, but it seems that the underlying cause of this change is also different, or is developing in these types of resistance mechanisms. So just to close this off, summary and question. So targeted DNA sequencing enables precision medicine using a liquid biopsy approach. Targeted DNA sequencing can be used to detect low-frequency mutations. This last example clearly shows that you can go down to maybe 1% or so of mutation frequency in your samples. But again, you need to have very powerful sequencers and very powerful bioinformatics tools in order to achieve this. Amplicon-based enrichment using multiplex PCR delivers a high level of specificity and uniformity that are required to overcome the low quality and quantity of liquid biopsy DNA. So again, you don't have a whole lot of DNA to begin with, and the DNA that you isolate is of low quality because it is highly fragmented. You need a method that is very specific and very uniform in order to look at these types of variants in your liquid biopsy sample. Redundant and overlapping amplicon design delivers the deep coverage required for detecting low-frequency mutations. Again, if you're looking at those low-frequency mutations, you also want to ensure that they get covered adequately, and having this overlapping and redundant design ensures that they get covered. And liquid biopsies are an efficient way to monitor disease progression and resistance. That last example was really very powerful in showing us how we can actually use liquid biopsies to monitor diseases. So with that, I think we can get to questions right now. I want to thank you all for joining, and let's get to the questions.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, how different is this approach than capture-based methods for enrichment, and which approach should I use?
There are two major ways by which you can enrich for your genes. First is the multiplex PCR and the capture-based approach. The main difference is the specificity and uniformity. So with the multiplex PCR-based approach, you get a very high level of specificity and uniformity, something that you cannot get with capture-based approaches. But the main advantage of a capture-based approach is when you have large targets. So if you have, like, let's say 300 genes or 400 genes that we consider as large targets, then the hybridization or the capture-based approach might be more appropriate. Multiplex PCR can be very powerful, very highly specific and uniform if you're looking at, maybe less than 200 genes. So that's when I would use the multiplex PCR-based approach. If I have something, 300, 400 genes or so, I would use the capture-based approach.
What types of variants can you detect with this enrichment approach? And is this for human DNA only?
So the types of variants with this approach, we can look at point mutations or SNPs. We can look at small insertions and deletions, something that is less than 40 bases or so, as well as copy number variations. So we cannot look at structural changes in DNA using this approach. And what we've developed here is, yes, for human DNA only.
Can I build a panel for hotspots only?
Yes, you can. So through the custom offering, you can actually define your targets the way you want. You can define the targets as exons. You can define them as only specific hotspots within those exons or introns. You can define intronic regions. You can define promoter regions. So you can use that approach to define whatever region you want, including hotspots.
What depths of coverage do you recommend?
The answer is going to depend on what types of mutations you're looking at. If you're looking at germline mutations, this is only my personal recommendation, I would recommend 200x for coverage. If you're looking at somatic mutations, like the ones that we see in cancer, I would recommend up to 1500x as mean coverage.
How much DNA can I get from a liquid biopsy?
So it's going to vary depending on the disease state, right? So if you're getting a liquid biopsy from a healthy individual, you might not get a whole lot of DNA from a liquid biopsy, maybe a total of 5 to 10 nanograms at most. But if you're looking at, or if you're using a liquid biopsy that has been taken from, let's say, a patient with advanced cancer, you're going to end up with a lot more DNA. So you might end up with 30 to 40 nanograms from a liquid biopsy.
And it looks like we may have time for just one more question. Are there differences of the results of a mutations profile when from one patient different liquid biopsies of origin can be taken, for example, blood and urine samples?
So will there be differences of the results of mutational profile? So that is a good question, and it will be different. Now I don't have I don't have an example here from the literature, but the difference, you're going to see differences depending on the sample that you use. If you use a solid biopsy, a biopsy basically that's taken from the tumor itself, versus looking at mutations that are associated with that tumor from urine or from blood, you are going to see differences because you're not going to get the complete mutational profile that is present in that solid tumor, right? The mutations that you see in blood or urine is going to depend on the ability of the tumor to shed those DNA fragments basically in the circulation. So if the tumor is not able to release DNA into circulation, you might not see those mutations in your liquid biopsies. So that's where the differences could come from. But there will be differences in the different liquid biopsies depending on, again, whether you can collect the DNA, whether the DNA has been shed from the tumor, and how powerful your sequencing approach is to look at these mutations.
I would once again like to thank Dr. Samara for his presentation. Do you have any final comments to share with us today, Dr. Samara?
Well, just as a final comment, the liquid biopsy approach using NGS to look at mutations is an approach that is becoming more and more popular. But I would definitely say that it has its limitations. It's not for every single tumor. It's not for every single case. You know, it's the amount of DNA that you get is a challenge. The quality of DNA that you get is a challenge. So if you really want to use a liquid biopsy approach, you know, just make sure that you have the powerful tools to sequence and the powerful tools to prepare your sample, and also the powerful tools to examine your results.
Thank you. I would also like to thank our sponsor, QIAGEN, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through April 1, 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time.